Hello everyone, welcome back to a perfectly normal day in Bio 2060. So, this is what we're going to be doing for this week, 4/6 to 4/13. Woo! So, we're going to have a couple papers that are going to kind of give you a little supplemental information from just what we're seeing with the lecture, since we can't do the super fun lab exercises. We're going to have to supplement them with other things. Yay! So, what we're going to be doing, this paper has a corresponding PowerPoint, so you can take a look at both of those, and then using the PowerPoint information, I'm going to be creating a quiz based off of this PowerPoint, and then also a lecture I'll be doing in a moment. And then we also have this paper, which is about kind of the effects of road salt and on the microorganisms surviving in those communities. And then there's a Salinity assignment that is based off of that paper. So you can take a look at that. All the information is in there. And then you're going to be posting that right here. So if you have any questions about that, feel free to email me. No problem. So here's our Lab 9 PowerPoint. First things first, let's get started. So this is about the different variables that have an effect on how microorganisms can survive in the environment and grow. So the first variable that controls their growth is through the environment they survive in. So the temperature, pH, osmotic pressure, or oxygen content. So the next thing is chemical methods. This is anything from just a chemical in there that is going to cause damage to them and not allow them to survive, or an antibiotic or antimicrobial peptide or molecule that is going to inhibit their growth or damage them in some way, inhibit their replication. Then we have the control by physical methods. So this is going to be heat. So if you heat something up really hot, so if you boil some water, radiation, so if you have really strong radiation, it's just going to destroy all the DNA, kill, and then filtration. The pores are going to be so small, it's not going to allow these larger celled organisms to pass through the holes. So, the next thing is, the very first thing that we're going to be talking about is how temperature can affect these organisms' growth. So, the majority of organisms, kind of the average organism, falls into the mesophile group, most pathogens especially. So they're going to be able to thrive in our optimal temperature range of 20 to 45. So we typically are going to be growing our organisms at around 37 degrees in our incubators in this lab. And mesophiles are going to really appreciate that. And so low temperature for them is about 5 to 20, and a high is above 55 degrees Celsius, all these. And for psychrophiles, they're going to be preferring a colder temperature. So that's going to be below 15 degrees about. And thermophiles and hyperthermophiles, so they love the heat, the higher temperature. And then hyperthermos just greatly in love. They're very passionate about the temperature. So they're going to be able to kind of thrive in these greater than 80 degrees Celsius environments. So they have a bunch of different techniques that they can utilize to survive in these different ways, different conditions. But we're not going to talk about that. Sorry. So what you would have done is you would have taken these plates and grown some things, but you didn't. Or you can't, unfortunately. So let me talk about pH. Neutrophiles, they're kind of your average organism, right? So most of them are going to be living in your range of about 6 to 8. So mainly, most organisms are going to fall between the 5 to 9 range, but neutrophiles are going to be even kind of right in the middle, as close to 7 as possible, typically. So acidophiles are going to be ones that prefer below 6, below 6 pH, and then alkaliphiles are going to prefer an alkaline environment, which would be above 8. So that's basic or alkaline, whichever way you want to call it. So fungi are going to be able to tolerate more acidic environments than most bacteria. So they're going to be acidophiles, and then archaea are going to be able to tolerate more alkaline environments. Okay, so here's a little chart that is going to show you the different pH numbers. So 0 is the most acidic, alkaline is the most basic, and then you're just going to look at the number, and these are some examples, and that'll tell you. So let's say in some tomatoes, which is down here, let's just say for the sake we got a real acidic tomato, it's about 4 pH, then it's going to have 10 to the negative 4 moles of hydrogen ions, and then it's going to have 10 to the negative 10 of OH minus ion, okay? So then you can see here that this is neither of these are very big, but in comparison, the 10 to the negative 4 is a lot higher than the 10 to the negative 10, right? So it's 10 to the negative 7 magnitudes larger. So here, it's 10 to the negative 7, here's 10 to the negative 7. This is pure water, right? Because there's the same amount of H compared to OH, right? Because water is H2O. So that'd be, here's your one H, and here's your other H, and here's your O. So that's kind of just how pH works. I don't want to spend any more time with that. So some more things that you would have done. We have these different tubes at different pHs, and we'd be seeing the growth of these different organisms and your unknown at the different pHs, but of course, you're not doing that. So the next thing is osmotic pressures. So these pressures are going to depend on the concentration of your dissolved salts and sugars in the water. So that's going to be your extracellular fluid. So compared to the inside of the cell, which is the intercellular fluid, that is going to create a concentration gradient between the inside and the outside, and that's going to affect the ionic strength as well. So isotonic is going to have a balanced ionic strength, and water isn't going to be moving in or out. But if it's hypotonic, then there's going to be a lower ionic strength, and the cells are going to swell. The water is going to be attracted inside, and in order to balance it out, and then eventually it can burst. And then with hypertonic, there's going to be a high salt or sugar in the media, and that's going to cause the cells to shrink, okay? So different microorganisms are going to require different salt conditions, right? So as we're talking about with temperature and pH, not all our microorganisms are going to want to survive in the same conditions. Some of them are going to try and specialize. So we have non-halophile, which is going to not require salt to growth, right? So then a halophile, based off these names and kind of figure it out, is going to require salt to grow, so just a kind of a small concentration. And then our extreme halophile is going to require a salt concentration of 10 to 20, much higher than this needs to grow. But then the halotolerant is going to be, so halo is salt, and then tolerant is going to be tolerating or tolerant of the salt. So it can tolerate some salt, but really it's going to not prefer very, very high concentrations of salt. The mechanisms that it's going to require to use to survive these conditions are not as efficient as just normal conditions. Okay. So, oxygen, as we know, I'm sure, is also a requirement for some microorganisms. Some kind of require the absence of it. So we have our strict obligate aerobes, which grow only in the presence of oxygen. Then we have our obligate anaerobes, which grow only in the absence of oxygen. So they do not want any. So aerobe is in oxygen, air, and then anaerobes is in no air. So micro aerophilic, so micro is in small, and then aerophilic, it loves the air, a small amount. So it's going to require a small amount of oxygen. If it has too much oxygen, it can't grow. If it doesn't have enough oxygen, it also won't be able to grow. So then we have facultative, which will use oxygen when it's available, but it can also grow when there's no oxygen. It's just not as efficient. So there are also aerotolerant anaerobes, which do not carry aerobic respiration mechanisms but can tolerate oxygen. Okay? So we have a couple different growth locations. So when we grow this tube, you can see that there's gas at the top. It's open. Air can come in and out. And the air does come in. And it has oxygen inside, as most air in our atmosphere does. Then that allows something with if there's no organism, it's not going to grow right uninoculated. But if it's obligate aerobe, it wants oxygen, then it will grow, but only at the top where there's oxygen. It's not going to be able to penetrate. Then a microaerophil will want a little bit of oxygen, so it's not going to grow at the top, but it will also not be able to penetrate down and grow down here because there is no oxygen. It's only going to be able to grow in this little sweet zone right here. So there's a solar system. This is Earth right here. This is Mercury, Venus, and then we got Earth and Mars. Now we're getting too far away from the sun, or too far away from oxygen. Then we have our obligate anaerobe, which is it's obligate. It's necessary for it to not have any oxygen, so it's going to be down here away from the oxygen. And then our facultative anaerobe can pretty much just survive wherever it wants. Lucky guy. Ugh. So for our exercise, we would have been growing some anaerobic organisms. Whoa! But unfortunately, we're not doing that. So if you're curious about the whole procedure and everything, feel free to look in the lab manual. We're going to use gas packs, and then we seal the jar shut, but alas, we cannot do that. Oh, here's an example of what it looked like, though. Just in case you're curious. 1 second. Oh, thank you for admiring that photo. So, okay. We don't need to worry about those other slides, but back to important stuff. Mannitol's salt auger. This is a type of selective media, and it's also differential. Remember, they can be both. So selective is going to have a high salt concentration, which is going to select for gram-positive bacteria. And then it's going to be differential because it has a pH indicator, phenol red, which is going to detect mannitol fermenting organisms, okay? So it has mannitol in here. If the organism ferments it, it's going to become more acidic. Then the pH indicator, phenol red, will make it turn red. Or it's the other way around. I don't know why I got that switched up. Okay. So the positive fermentation is going to be yellow, and the negative fermentation is going to be pink. Okay. Oof, that was rough. Moving on. Let's try and ignore that. Wow. And with that, we're all done. Almost made it to the end without doing something terrible. Well, you gotta try your best. Sometimes you fail. Sometimes you succeed. today, well, we're not done yet. Biogeography of soil bacteria. This is the PowerPoint presentation that I posted on Blackboard. So, soil bacteria, just a little background information on them. Actually, I'll do a little background information on this PowerPoint real fast. So, if you're listening, you can fast forward a couple seconds and skip ahead, or you can watch. So, I did this for a biogeography class with one of the other instructors for this lab, Robert. And we created this as an assignment for that graduate course last semester. And I thought it kind of tied in with what we were talking about. And I thought it might be a little supplemental information for what we're talking about. So, moving on. Soil bacteria, the most abundant microbe in soil. It's going to serve many purposes: nitrogen fixation, nutrient cycling, degradation, remediation. And yeah, I'm not going to go over everything. I'm so sorry. This is a lot of stuff. A lot of stuff. But it's based off of this paper. A high degree of environmental covariates control the biogeography of soil bacteria. Yeah, quite a mouthful, I know, geez. But it's pretty interesting. I thought it was a pretty clear read. I'd like you to just take a look at this PowerPoint, and it's pretty self-explanatory, most of it. I just have a couple things I want to go over. So this 250 meter soil grids is basically just a large 250 square meter grid system that kind of puts all the different soil properties kind of collected from that area. I'm pretty sure that it was all just statistically estimated based off of samples around that area, but you can Google it if you want more information. So we have a couple different figures in that paper, and all this is different. All this is showing is just the distribution of the bacterial richness in different areas. So the bacterial richness is basically just the amount of different, kind of the biodiversity, okay? So the higher the richness, the more biodiversity of the bacteria in that area. So they just took a bunch of different samples, and then they were able to figure out that certain areas had higher levels of richness. And then they compared that to different variables that we're just talking about, right? So pH, water content, climatic water content, and then also you can kind of get an idea. So these area right here, 400 about, right? So it's a dark blue. So that's going to be a lower biodiversity, and then down here, these areas of red are going to have higher amount of bacterial richness, higher diversity, right? So just take a look at this PowerPoint and take a look at the paper, and then I'm going to be quizzing you on these two PowerPoints. So if you have any questions about that, feel free to let me know. And with that, we're all done. Woo!